click the start button.
All right. See you on the other side.
I know. We'll slowly have people trickle in.
So we should mute and turn off our cameras, I assume.
Yeah, if you want to for now, you can mute and turn off your cameras, and then once we do the update, I will you guys can turn your cameras back on and we'll have some fun. Really need to make this like have some music or something. I keep saying that, and I'm not gonna sing, so sorry, no singing today.
That would be fun.
Fun for some, entertaining for others.
Yeah, you need a theme song, you know?
Well, it's just so different with in person, right? You have those side conversations, and as people come in, and then here it's just kind of like dead silence.
Well, it's also helpful to have the music to know that you're actually connected, or as if, because it's so
that
often.
too. That too. All right. Well, people are trickling in, which is good. I think we'll just go ahead and get started as people join us. But thank you, everyone, for attending this. This session from uniQure, who will be hearing about some HD research updates. We'll be watching a somewhat quick video that will give an update, followed by a Q&A with Dan Leonard, who works over at uniQure, overseeing their patient advocacy function, as well as Sarah Ying, who is the director of clinical development at uniQure. And so what I always mention is, if you have questions, feel free to put it in the chat or use the Q&A function that you should see below you. And don't hesitate to ask a question. You can also make it anonymous if you feel comfortable doing it that way. The one thing I will say, and I mentioned this in the Roche one that I presented or that I moderated, is that, you know, if you are participating in the study or a family member is, and you have specific questions, please reach out to your trial site coordinator for those. It's very important to, you know, speak to them when it comes to anything regarding the specific trial. So just wanted to kind of reiterate that piece. But without further ado, I'm going to share my screen and we'll get going. Sarah and Dan, can you see the video?
Yes, I can.
Perfect. Awesome. All right. I'm going to go on mute and we're going to get a quick update from this video, and then again followed by a Q&A portion. So if you have questions, don't hesitate to ask them throughout this, and we'll do our best to get to each one.
Hi, I'm Dr. David Cooper, Vice President of Clinical Development and the HD Program Lead at uniQure for the AMT-130 study. It's my pleasure today to be able to present updates on the HD GeneTrix-1 Phase 1/2 clinical trial of AMT-130 in early stage HD. And we have a lot of updates to present on the trial. It's my pleasure to present on behalf of the uniQure team, our steering committee, our investigators, and our investigative sites. These are my disclosures. So let's start with just what's gene therapy. The basic concept is gene therapy is a technique that uses genetic material to treat or prevent disease. So there's a capsid, which is the package, so to speak. There's a DNA expression cassette, that's the instructions that allow the cells that are targeted to continuously produce the therapeutic molecule. So we all know about Huntington's disease. There's this repeat in the Huntington DNA that ends up causing a prolonged CAG repeat in the exon 1 in the mRNA, which leads to an expanded polyglutamine tract, which leads to protein aggregation and neuronal degeneration. So what's our target with gene therapy? So our target is to target exon 1 mRNA and block the production of the protein. So let's go in a little more depth. What is AMT-130 and how do we deliver it? So AMT-130 or AAV5 MyHTT, consists of an AAV5 capsid. And AAV5 is something that we use across multiple of our platforms. And then there's an expression cassette that codes for a microRNA inside. This is a replication deficient AAV5. The microRNA targets the toxic Huntington exon 1, and it uses our uniQure technology. How is it administered? It's administered in a one-time intracranial stereotactic administration into the striatum, namely the head of the caudate and the putamen. Proof of concept comes from a very extensive program of animal modeling. We started with cultured human neurons, looking at efficacy and safety. We've just explored AMT-130 in HD rats in 4 types of HD mice, where we've looked at distribution, efficacy, and safety. And ultimately, we were able to translate this into large animal models, the transgenic mini pigs, as well as non-human primates, where we looked at distribution, efficacy, and safety, and particularly at the MRI-guided stereotactic administration procedure that we're using in the clinical trial. So let's look at a little more detail at what exactly is intrastriatal convection enhanced delivery. So this is a very deliberate approach where we're using intraoperative MRI scanning to target the head of the caudate and the putamen, shown here, along with the lateral ventricles in this cutaway of the brain. The goal is that during this operative procedure, there are 3 separate injections on each side done in pairs, first to the anterior caudate, the head of the caudate, then to the anterior putamen, and then the posterior putamen. Each of these are 500 microliters, and then with convection enhanced delivery and real-time monitoring with a little bit of trace amount of gadolinium, we're able to follow as these injections are done in real time on the MRI scanner. With antegrade and retrograde transport and other transport mechanisms, we do know from animal models that we do get broad cortical and other brain structure distribution from an injection in the striatum. So that's why we think we'll have a broad effect. The goal of treatment ultimately is to slow disease progression. And based on regulatory feedback and other considerations, the time that most of disease modifying treatments have looked at is the early manifest period after the disease progression really starts. Our goal is to slow that progression, if not stop that progression. So the AMT-130-01 phase 1/2 study, or HD-GeneTrix-1, this is the first FDA-approved clinical trial for gene therapy for patients with HD. As a first-in-human trial, the primary objective of the trial is to look at safety and tolerability of AMT-130, an HTT gene-lowering therapy in early manifest HD. The secondary objective is to test the duration of AMT-130 persistence in the brain. And the exploratory objectives are to examine the effect of AMT-130 on biomarkers of HD and to assess HD progression and response to AMT-130 treatment. So here's where we get into some of the particular updates to the program. So this is a double-blind, randomized imitation surgery or sham surgery controlled study. We're testing 2 dose levels of AMT-130, 6 × 10⁻¹² and 6 × 10⁻13 genome copies per patient. So this is equivalent to 50% lowering of HTT in the striatum at the lower dose and 25% in the cortex. And at the higher dose, 75% lowering in the striatum and 50% in the cortex. As I said, it's administered by a one-time bilateral stereotactic neurosurgical administration to the caudate and putamen via MRI-guided convection-enhanced delivery. The sham procedure consists really only of skin incisions and anesthesia. The patients remain asleep in the MRI machine for the same amount of time so that they think they've had the surgery, and they wake up with the same skin incisions from the very small puncture wounds that are used to do the burr holes and administer the therapy. The blinded treatment period is now 12 months, not 18 months, so this has been updated. The total follow-up is 5 years for the patients only that have received AMT-130 treatment, and our plans are to conduct this in 9 to 12 HD centers in the US, including 3 of them that will be performing the surgery. So the right shows the schematic with the initial period being 12 months of blinded follow-up and then the long-term follow-up 4 years. In total, this is a small phase 1/2A study. We expect to enroll 26 patients in the study. The low dose group of AMT-130 has 6 patients. The first 4 of those are, so here we go into the organizational structure of the study. In total, we're enrolling 26 patients in the study. There will be 6 patients exposed to low dose, 10 patients exposed to high dose, and 10 patients who undergo the imitation or sham surgery. The crossover will be implemented after the FDA and the DSMB review preliminary safety and efficacy data, and this is a change that's now been incorporated into the protocol. AMT-130 will then be offered to patients completing the 12-month visit that continue to meet the original eligibility qualifications of the study. So now the details of inclusion and exclusion, and here's where some of the experience we've had early in the study has helped us to refine the criteria for inclusion and exclusion. The age for inclusion and exclusion remains 25 to 65 years of age. Patients have to have a diagnosis of early manifest HD based on UHDRS with a TFC score of 9 to 13, which is updated, and either a UHDRS DCL of 4 or a DCL 3 with either a positive response to the UHDRS question 80, which is the multidimensional classification. Are you 99% sure that the motor, behavioral, cognitive, and functional symptoms are suggestive of HD? Or the DSM Diagnostic and Statistical Manual 5 criteria for a cognitive disorder, which is the Movement Disorder Society Task Force criteria. The number of CAG repeats is now 40 plus, so full penetrant mutation. Originally that was 44. The volume requirement, which is very key since we're doing administration into an atrophic striatum, is a putamen volume of greater than or equal to 2.5 centimeters per side and a caudate volume of 2.0 centimeters per side. All HD concomitant medications need to be stable for 3 months prior to screening. And this includes medications for movement symptoms, motor symptoms, cognitive symptoms, or behavioral symptoms. There is no longer an exclusion criteria for any chorea-based medications. Key exclusion criteria are the presence of an implanted brain stimulator, ventricular peritoneal shunt, or other implanted device, and any brain or spinal pathology that may interfere with the surgical delivery of AMT-130 or represents a significant neurological concomitant disorder. Previous gene therapy and other investigational agents are also excluded, and as well any patients who have a contraindication to lumbar puncture. So these are the revised criteria in the current protocol on clinicaltrials.gov based on the initial experience with screening and how we've revised them to this multidimensional classification of early manifest HD. What are our endpoints for the study? Well, the primary endpoints, this is primarily a safety study, so we're looking at adverse events. We're looking at clinical parameters, which include MoCA or the Montreal Cognitive Assessment, laboratory parameters in blood and CSF, changes in brain structure and functioning on MRI, AAV vector shedding, and immune response to treatment. Secondary endpoints from the perspective of monitoring PK are levels of vector DNA and microRNA in CSF over time. The exploratory endpoints include HTT protein levels, mutant and total, in the CSF, neurofilament chain light, NFL, in CSF and serum, clinical endpoints including UHDRS, Q-motor, HD-CAB, imaging endpoints including volumetric and functional and MR spectroscopy, as well as cognitive endpoints, including HD-CAB. So where are we? We were very happy that we received orphan status from the FDA and EMA. In June, we were very pleased to announce that the first 2 patients were treated in the phase 1/2 trial after a delay due to COVID-19. In September, we recently announced that the Data Safety Monitoring Board, or DSMB, recommended proceeding with study enrollment with no significant safety concerns. And earlier this month, on October 13th, we announced that the second 2 patients were treated in the phase 1/2 trial. So we've made a lot of progress since the last HSG meeting a year ago. So enrollment is ongoing at sites in the US. More information about the trial can be found on clinicaltrials.gov. And here's the clinical trials number, and through patient organizations. We also have an email and a phone number that people can call for more information. We're very pleased at the moment to have 9 sites that are activated, the last 2 of which will be soon to be recruiting. But we very much appreciate all the support of these sites, the principal investigators, the study coordinators, and both the blinded and unblinded study teams that are working to support the study at these centers. I want to just thank the uniQure team, which spans everything from the research group to manufacturing, clinical development, assay development, et cetera, all reflected here. We're very thankful for the support of our steering committee, who has helped across our trials program, our neurosurgical committee, as well as the HD community and many collaborators. Thank you very much.
All right, I think that's that was it for the video piece of it. Sarah and Dan, do you want to come back on? And cool. Well, that was, very informative of understanding, the update, and maybe Sarah, you can kind of touch upon, or Dan, either one, just touch upon kind of where you're at now. Are there any updates that the HD community should be aware of when it comes to recruiting or even participating in a study like this?
Sure. Why don't I go first, and then I'll hand it over to Sarah, and I'll just reintroduce myself. So Dan Leonard, Senior Director of Global Patient Advocacy at uniQure. I've been at uniQure since 2016, so I've been at the company for quite a while. And I would just right away out of the gate want to say thank you so much to the HD community, to HDYO, and all of the patient organizations and patients and family members that we've worked with over the past, since I've been at uniQure for the past 5 years. It's kind of hard to believe that we are sort of at the beginning of a phase 1/2 clinical trial, which is the earliest phase of clinical trials. Yet this program, I've been working on it for 5 years, and it was happening many years before I ever joined uniQure. So it really is such a long journey, and it's a journey that can't happen without the patient community. And particularly in a trial like this, where it's so novel, it's so new, folks that step up to volunteer for the trial are without a doubt incredibly brave and heroes, truly. So just a deep thank you to everybody for the support and the involvement. I do want to note that in the development of the protocol for the clinical trial, we did solicit feedback from patients and from the patient community. So we did an HD COPE session with patients and caregivers from Europe and from the US, which was incredibly helpful and informative and certainly shaped the protocol. Having said that, when you're doing such a novel trial, you are learning along the way as well. And there's certainly a process where you're fine tuning. And so you heard a little bit of that from David in the video, where, there's, so again, some fine tuning with the inclusion/exclusion criteria. And then the last thing that I'll say is that, as you probably noticed, that video is a little bit dated at this point. That was from last fall. So there's still a lot of the factual information is still very good. So, what is gene therapy? What is gene therapy for HD? What is this particular therapy? How does it work? How is it delivered? All of that has stayed the same. But there are certainly some updates from the execution of the trial that Sarah will provide. And I think, and the last thing is that we are working on an updated video. So hopefully there will be yeah, in the future, we'll have something that's a little bit more up to date. So we almost had it done for this, just didn't quite make the deadline. But we are lucky to have Sarah here, who will talk about some of the updates from the time that video was recorded until now.
Thanks, Dan. Hi, I'm Sarah Ying. I'm a neurologist working in the clinical development side of things, and it's just really exciting to be part of this program, which, as you know, it's the first gene therapy for Huntington's, and we're learning along the way, along with everybody, and so we really appreciate the all the enthusiasm and effort that everybody's put in, and we thank particularly our participants. And so and since then, we've started, we've already treated our first 4 patients, 2 of which have received the treatment, 2 of which have received the sham surgery, and we're currently working on the next set of 6. And so this is the low dose cohort, and once we're done with this first cohort, we'll be moving onward. We're excited to say that we have there's a couple more sites that are just opening up. So all of you who have been looking, Boston, yay. Beth Israel Deaconess has just opened up as a site, and also Johns Hopkins in Baltimore has just opened up as a site. So our 2 East Coast B cities, and we are and we will probably be opening up a couple more sites in the near future as those come online. So happy to entertain any questions if there are any about what's been happening, but we're just excited. We're all blinded, so there really are no updates as to what's happening study-wise, but we really appreciate your partnership with us in keeping that blind, and just as Seth was mentioning earlier, that it's just really important that that all of your thoughts are kept, within the family and to yourself, so as not to jeopardize the scientific aspects of the placebo aspects of this trial. And so again, thank you so much, and we're excited to be moving forward, and we're excited for all the questions that people have had, and we're just happy to be here.
Yeah, no, I mean, it's a very exciting time. As you both mentioned, gene therapy is new in the HD community. It's actually fairly somewhat of a new concept just in general, right? In the rare disease space. And I think it's important to understand the difference, right? Because this is supposed to be that like one and done type approach.
Mm-hmm.
And so that being said, I mean, maybe again, I know it's very early on, but when you think about it from that perspective, how can the HD community kind of play an active role, to make people more aware of this approach, but also kind of that access piece of it. When you look at other rare diseases that may have gone through the gene therapy route, like that, I know that access can be sometimes challenging. So maybe you can kind of hit on that piece of it.
Sure, and from an access front, so that's so Seth, you're absolutely right. I mean, gene therapy is still somewhat of a new modality. Right now, there are only you can count the number of approved gene therapies on one hand right now. Mm-hmm. And that it's been a really long time coming. People have been talking about gene therapy for decades, 3 decades, literally. And yet we're just seeing the first gene therapies becoming widely available. So the first gene therapy that was approved in the US was for a rare form of genetic blindness. And then shortly after that was a gene therapy for another rare genetic disease called SMA, spinal muscular atrophy. In fact, uniQure actually got the first gene therapy approved in Europe. And that was quite a number of years ago. So there was a little bit of an outlier. uniQure did have a gene therapy approved in Europe back in, I think, 2013. again, for another rare genetic metabolic disease, but wasn't really widely known, tiny patient population. And so that wasn't very well known, but we are starting to see gene therapies get approved and become available, and we're going to see quite a few more. There should be a gene therapy approved this year for hemophilia A, and now you're starting to get into disease communities that are getting larger and more well known. So you're going to start to hear about this more. And we've seen challenges with access already in the 2 gene therapies that have already been approved. Part of the reason for that is there's just not a good model that exists for how to make these accessible. As you can imagine, the difference between a chronic lifelong therapy, a therapy that you would be taking for your whole life, versus a potentially one-time therapy, how you would pay for that is going to be completely different. With a chronic therapy, you're sort of diluting all of those payments out for the course of somebody's lifetime, right? For a one-time therapy, it's kind of concentrated into this one time. But if you took all of those payments that are stretched out over somebody's lifetime and you condensed it into one payment, that would be too much for any person to pay on their own. So how do you do that? And you'd say, well, there's all these different ways you could space the payments out, which sounds like maybe that would make sense. But when people are paying through their insurance and they might switch insurance companies, every few years, how does that work? Who's responsible for the payment? So it's actually pretty complicated. There are scenarios that have been worked out with the 2 therapies that are approved right now, and they're again, it's a learning process. So there are new ways that are being looked at and trying to figure out what has worked for those 2, what hasn't worked, how can we optimize it, how can we make it better. But I would say, your question was, how can people in the patient community be involved and engaged in that? And I would say the most effective way would be to work through your local patient organization, wherever you are, whether it's in Europe, whether it's in the US. Without a doubt, there are organizations that are engaging both local and national governments around issues of access to advanced therapies and emerging therapies. And the more patients and patient organizations raise the awareness of this with politicians, governments, and regulators, the more that it's going to really force their hand to come up with solutions. In the U.S., it's a little bit different because we have obviously a lot of private payers.
Yeah, that's a tough part. And I was just gonna say, you'd think it could be kind of like when you're buying like a home or like you're paying off your college loans, where it's like you have these monthly payments based off of that piece of it. But it does, it is intimidating, right, to look at these larger prices, but then trying to figure it out, how do you expand that, right, that long-term outcome, and then trying to break it down month by month and whatnot. But I think,
yeah, I mean, it really would be, in an ideal scenario, it's really going to be the insurance company and or government payer that's going to have to space those payments out. But even for them, it's intimidating, right? So there's the individual, and obviously you want to minimize the impact on the individual. And then there's the payer, whether it's, again, a private payer or a government payer. I mean, even the private payers and the government payers are like, whoa, how are we going to do this? So just suffice it to say that everybody's committed to making it happen. I mean, we as a company it would make no sense to spend 20 years developing a therapy that then nobody could use because it wasn't accessible. So we're absolutely committed to making sure that it's accessible. But I will say that there are still, without a doubt, I'll be very, very honest, there are still details that are going to have to be worked out, and it's a work in progress. But absolutely advocating, through patient organizations, going to, state Hill days where you can partner with a patient organization and go to Capitol Hill, virtually, and lobby your local representative. That absolutely is a powerful way to be engaged and involved in the process. And it actually does make a difference. It truly does. So you can truly be a part of what happens.
Yeah, absolutely. And I think that's important is for members of the community to advocate for these and really help make, members of Congress or people kind of legislation challenges that could occur, right, when it comes to pricing, and trying to just understand that piece of it.
Absolutely.
Ultimately, yeah. Ultimately,
it's a state level too. Just to be clear, right? So there's lobbying at the federal level, and without a doubt, there are Capitol Hill days where you go to Washington D.C. or virtually go to Washington D.C. But there's also state Hill days as well, and those are just as important. Absolutely,
yes, I totally agree. I wish we had more time for this conversation and to also just kind of discuss the next steps, but I believe we are a minute over from what I'm looking at. But we do appreciate the update from uniQure's side. It is very exciting. Of course, we wish we were all in person, and hopefully one day can be in person to have more of these important discussions. So thank you, Sarah and Dan, for just being here, discussing some updates, answering some of these questions. And for everyone else who is an attendee, take a look at, the next upcoming sessions. We have a session track 1, which is just a Voyager HD research update. And then on track 2 is a panel on testing negative. So check those out, and thank you both again. This was great.
And please visit our virtual booth. There's a lot of information there, and
yes.
It might even be, so definitely check it out.
Yes, the more information, the more knowledgeable, the better we can prepare ourselves. So good call, Dan. Take care, everyone.
Thank you. Take care.
Thanks,
Bye-bye.
everybody.